Greetings from Sydney, Australia. My name's Colin Clifford, and I'm going to be talking about what brain decoding can tell us about consciousness. First of all, I'd like to thank Richard Brown for inviting me, and I'd like to say that this is my first attempt at making any kind of screencast, so I hope it's reasonably watchable. I'm going to adopt the punk rock ethic of trying to do everything first take without any overdubs or clever stuff. If for no other reason, then I'm not sure that I actually have the technological capability to go back and edit this once it's made. So what I'm going to do is talk over these PowerPoint slides that I've presented, and you should also be able to see from time to time my mouse pointer highlighted on the screen. So to begin with, what would we like brain decoding to tell us about consciousness? I think we can distinguish two areas here. One is the contents of consciousness, that is, what's in my mind at the moment, what am I seeing or thinking. And another qualitatively different issue is the neural signature of consciousness. What is it that distinguishes conscious from non-conscious processes? And can we use brain decoding to work towards an answer to this question, potentially see whether there is conscious activity in the brain. So, to begin with, I'm going to talk about what brain decoding is, give an idea of what's new about it, how it works, and to discuss briefly the idea that it's mind reading, which is a tag which has been popular in the media. I'm conscious that there's a lot of busy people out there, and I'm also conscious that I'm creating a larger and larger file all the time that I'm talking, so I'm going to try and keep things brief, but hopefully give an overview, and if people have further questions, then I'll be online over the next couple of weeks, and we'll be happy to respond to them. And this talk could expand into a whole lecture series. I need to be focused, so I'm going to focus on specifically vision and functional magnetic resonance imaging. Vision because it's what I know about, but also because it's an area in which this technique has been profitably applied. And functional magnetic resonance imaging, again, because it's the area I know about. It's also the primary technique to which brain decoding has been applied, largely because of its high spatial resolution, although in principle brain decoding can be applied to other measures of neural activity. So what is brain decoding? Well, in short, it's multivariate pattern analysis of brain activity. What's new about it? Well, traditionally, people have studied activity at a univariate level. So we might look at activity in a voxel, which is like a three-dimensional pixel, looking at activity in a small region of the brain, maybe a couple of millimeters on each side, or an aggregate of voxels. We might define a region of interest on anatomical or functional grounds, and then look at the activity in that voxel or that region. And then typically we might present stimulus A and stimulus B, and we'll look at activity, some measure of activity, as a function of which stimulus is being presented, and see whether we can observe statistically significant difference in the level of activity. This level is a univariate measure. Now, that's all very well. For example, if we wanted to identify a region of the brain that responded more strongly to faces than houses, then we could use this sort of univariate measure. But what about if we wanted to see whether that region that we'd identified that responded to faces actually contained information about, say, identity or expression or certain attributes of faces? Now, there's no reason to think a priori that one face should, or one emotion should necessarily produce more activity in that area than any other, but we might expect that we'd be able to discriminate those identities or those emotions on the basis of the pattern of activity in that area. And that's where we look at multivariate analysis. So say that we have a region of interest composed of a number of voxels, then we can look not just at the aggregate activity in that region, but we can look at the pattern of activity across that region. And so the response to stimulus A and the response to stimulus B in these collections of hypothetical voxels might give us a sort of a detailed pattern of response to a particular stimulus. So what this allows us to do is to take cases where univariate measures are uninformative, there's no net difference between the response to stimulus A and stimulus B, and see whether we can still pick out stimulus-related information. Now, how do we do this? Well, we need some way of learning the difference between stimulus A and stimulus B. So we need to employ a learning algorithm. For example, support vector machines are a common form of learning algorithm from the machine learning literature. What they do is, when presented with a training set, you present them with a set of patterns of activity that have been recorded in response to stimulus A, a set of patterns recorded in response to stimulus B, and effectively you tell the algorithm, these are the patterns from stimulus A, these are the patterns from stimulus B, learn the difference. And these algorithms are smart. Typically they can learn the difference, but the question is, can they generalize the difference from training to test data? Now this is an important point, because if we want to be sure that the learning algorithm is actually learning the difference that we want it to learn, rather than some other spurious incidental stimulus difference, then it's no good training and testing on the same data. It's important to see whether the discrimination that we've learned generalizes to a set of data on which the algorithm hasn't been trained. So typically studies employ a leave one out process, where there might be, say, 10 runs where stimulus A and B are alternated, and you might train the learning algorithm on the responses from 9 runs and then test on the 10th. And then you can repeat this process 10 different times, each time training on 9 runs, testing on the 10th. And that way we can measure the performance of the algorithm on the basis of its generalization from training to test data. And we can see whether it performs significantly better than chance. Now, assessing significance is not always trivial, but in short, we can perform a technique called bootstrap relabeling, where we shuffle the labels on the training data and see what levels of performance we might expect by chance. If we perform, say, 1,000 of these random simulations, then we get a distribution of performances, and we can see how likely, just by chance, we would be to observe performance at the level that we see with the real data. So I've said that brain decoding is multivariate pattern analysis. That's what's new about it, going from univariate to multivariate measures of brain activity. Now, is this in any real sense mind reading? Well, first of all, where does this mind reading tag come from? This is not something I've traced the history of, but I think it lies in the fact that multivariate pattern analysis typically employs a train and test prediction-like framework, where if you're training on a set of data, then you're using the rule that your algorithm has learned to predict what the subject was seeing in the test data. So you're training on 9 runs, and then you're predicting what was being presented in the 10th run. So in a sense, the algorithm is saying, on the basis of what I've learned, I think this is what you were looking at, this is what's going on in your mind. So while I can see how this sort of label might have come about, in the sense that brain decoding is mind reading, isn't any measure of neural activity mind reading. In principle, one could employ the same train and test prediction framework to any measure of neural activity. Clearly with univariate activity, you don't really need to do that. You can just say with a univariate measure, we get a bigger response to A than to B. The difference when you're using a multivariate approach is there's no such thing as a bigger response. If you want to say that the responses are a difference with multidimensional data, then you need some way of establishing that difference, and that's where the train and test framework comes into play, and that's where you then have the trained algorithm predicting what's being seen on test runs. A final point, this is nothing that I think anybody has actually claimed, but I think it would be a reasonable misunderstanding. The fact is, brain decoding tells us nothing about the language of the brain. If you're thinking of something as mind reading, then maybe in a sense you think that the algorithm is reading the language of the brain. But because the decoding is occurring at the level of voxels that are part of the data acquisition process and extrinsic to the brain itself, what's being learnt by the algorithm does not relate in any simple way to the way that brains transmit information, to the way that regions communicate with one another through neuronal spikes. It's evidence that that information is represented in that region of the brain, but it's no closer to the actual neural processing than that. So, magnetic resonance imaging is the technique that is predominantly employed in these brain decoding studies, and I must acknowledge Judy Cullen's website at University of Western Ontario for this nice illustration. The machine is the same sort of MRI scanner that we have at your local hospital these days. MRI studies brain structure. What we look at is the unchanging parts of the signal coming from a subject's brain. fMRI, functional MRI, studies brain function. What we're interested in is what changes over time. And to do this, we use the BOLD activity, the blood oxygen level dependent signal, as a measure of neuronal activity. So they're obtained in the same machine, the same subjects, in the same session, just in different scans with different sequences. So we can co-register these functional and anatomical data so that we can have high resolution maps of where changes in activity are occurring within a subject's brain. And specifically when we're studying vision, typically prior to the experiment proper, we can map out the early visual areas of each subject's occipital lobes on the basis of the response to a rotating wedge stimulus. We use a rotating wedge stimulus because we know that there are multiple maps of the visual field in the subject's occipital lobe, and so from the position or the phase of the rotating wedge stimulus that gives the maximum response, we can work out where in the visual field a given voxel receives its stimulation, and thereby we can create these pseudo-color maps of the response of visual cortex to different parts of the visual field. So this is a left hemisphere of one subject, and so you have a representation of the contralateral and right hemifield of visual space. And we can use these responses to delineate the borders between the various regions. Certainly V1, V2, V3 are clearly divisible on the basis of these kind of retinotopic data. So once we have these preliminaries out of the way, we can now define regions of interest. We can say these voxels constitute V1, these voxels constitute V2, these voxels constitute V3, and now onto a couple of pioneering studies in brain decoding, and they're really only 5 years old. There are two here that are actually very similar, performed in parallel, independently. One by Haynes and Rees, and one by Kamatani and Tong. And both of them were looking at orientation coding in the early visual areas. The processing of visual orientation is something that is known from the mammalian electrophysiology literature to be a property of visual cortex. You don't see orientation selectivity in neurons earlier in the hierarchy than primary visual cortex. And what both of these studies show is that it's possible to discriminate orientation using this brain decoding method as early as primary visual cortex. So we have from the Haynes and Rees study the generalization performance for areas V1, V2, V3 in discriminating between leftwards and rightwards tilt. So chance performance would be 0.5, perfect would be 1. Because fMRI data are noisy, you never see anything close to perfect performance, but they across areas V1 to V3, so, the algorithm was decoding test data on which it hadn't been trained at between 70 or 80% correct. Kamatani and Tong employed a slightly different procedure where they actually trained and tested on 8 different orientations rather than just one, and then they looked at the algorithm's performance on the test data. And what they noticed, first of all, was that there was very good performance, that the majority of responses were correct, and they also noticed that when there was an error, it tended to be to a nearby orientation. So that's why you get these long and narrow tuning curves, if you like. So these are the responses of the algorithm to a particular test pattern. So most of the time it's classified correctly. Occasionally it's mislabeled as a neighbouring orientation. So how does this work? There are two senses in which one can ask that question. One is, well, how does it work in terms of what's the algorithm that you employ? But then the other is in terms of, well, what's the neuronal basis? What is the algorithm actually using in order to be able to perform the classification? And this is something that's I think still a subject of debate. And let me outline really the continuum of possibilities. There must be some information in the response of the region that is able to discriminate the activity, and so, to discriminate the stimuli. And the response of the region is a pattern of responses across voxels. So if there's a pattern of responses across voxels, then individual voxels must have some information about the stimulus, maybe very small biases, such that although averaged across the region there may be no more response to one orientation than any other, at the voxel by voxel level each individual voxel might have its own characteristic bias, and when combined in an intelligent way by learning a pattern, the algorithm can make this discrimination. So at one extreme it might be that there's actually a statistically uniform distribution of neurons across the cortical surface, and we simply have by chance biases in the sampling of this distribution, simply because we're only sampling from a few neurons, although we're probably talking hundreds of thousands in a given voxel. So according to this view, the classifier wouldn't necessarily, successful classification performance would not actually require any spatial structure in the neuronal representation. A more conventional view is that successful classification performance is evidence of some sort of spatial structure. And then the question would be, well, at what scale is that spatial structure evident? Is it at a subvoxel scale, whereby there are biases or inhomogeneities in the distribution of neurons with particular tunings across the cortical surface, and then although these are at a finer scale than the scale sampled by voxel, they're sufficient to produce small biases in response to that voxel. That's something I'll illustrate in a moment. Or alternatively, it could be that the pattern of bias is at a spatial scale much coarser than a single voxel. For example, perhaps a quadrant representation of the visual field. I'll illustrate these two latter points. I think the chances are that the first possibility is not going to be able to produce significant biases, but I don't think we actually have the data yet to definitively rule out this possibility. So I don't think we can yet say categorically that successful classification performance is evidence of spatial structure. So the idea put forward by Jeff Boynton and others is that decoding of orientation in V1 might work in the following fashion. We've seen from optical imaging studies on primate visual cortex and cat visual cortex that the orientation preference of neurons tends to be organized in a systematic pattern, such that neurons with similar orientation preferences are grouped together in orientation columns. If this is the case, then there's some structure within each voxel in the way that orientations are represented. So here this color code translates into the preferred orientation of a given column of neurons. And if one is to then look at the distribution of orientation preferences within each voxel, what one finds is that there's a broadly equal distribution, but that just by the virtue of the fact that we're sampling only a finite region of the cortical surface, some regions, some orientations are represented preferentially over others. So for example, there's more vertical tuning, the light blue, in this lower left-hand voxel than there are in other regions, than there are at other orientations. Whereas if we look at a different voxel, we might find, for example, if we look at the top left voxel, there's more green, which is rightwards tilted orientation. So these are small but potentially systematic sampling biases of spatial structure at a scale finer than a voxel. And what a classifier could in principle learn is that if there's a strong response from this given voxel, it's more likely that the orientation is vertical. If there's a strong response from this one, it's more likely that the orientation is tilted slightly rightwards, and so on. Although each of these biases is small, if we have, say, 1,000 voxels in V1, then the information can be intelligently accumulated by a classifier, and potentially it can learn on the basis of this biased sampling to discriminate different orientations. However, it's possible that there's an altogether potentially less interesting explanation for why classifiers are able to decode orientation in V1, and excuse me, this is something that was first demonstrated by Sasaki and colleagues from Roger Tutell's lab, and that is that there's a systematic bias towards radial orientations in visual cortex. So if we fixate on a central point, then if we present a horizontal grating, then the orientation of that grating is approximately radial along the horizontal meridian. Similarly, rightwards grating has radial orientation along the rightwards meridian. And so to see what effect this has on the response of the early visual areas, I've presented below a contrast between the response to leftwards and rightwards oriented gratings from a PhD thesis of my former student Kylie Seymour. And what you can see straight away, if you take a coronal section through the occipital lobe, is that the regions representing the two quadrants of the visual field where orientation is primarily radial for a rightwards oriented grating give a much stronger response to that grating than they do for the leftwards oriented grating, and conversely the other two quadrants give the bigger response for a leftwards oriented grating. So what we have is this nice hot and cold map at the back of the head, which is really picking out orientation on the basis of the activity of particular quadrants. Now, if you take the whole of primary visual cortex, for example, as a region of interest, then because that includes activity from each of those quadrants, you wouldn't expect the whole of primary visual cortex to show any univariate bias. However, the spatial scale at which that bias is evident is at the level of a quadrant, let's say a quarter of V1. So it's clearly a much coarser level of representation than a voxel. And so mind reading in this circumstance is really pretty trivial. I think you can mind read. You could mind read if I showed you an image with activity in these two quadrants. You'd know straight away that it was a leftwards grating. If it was in these two, it was a rightwards grating. You wouldn't need any fancy machine learning algorithms. So to investigate whether it's that simple, whether these very coarse anisotropies in the representation of orientation in visual cortex are what underlies brain decoding, we did an experiment, Damien Mannion, my PhD student, Scott MacDonald and myself, where we presented stimuli that were everywhere oriented 45 degrees from radial. So what we have is a clockwise and a counterclockwise spiral. Now these are actually mirror images of one another, and we wanted to see whether brain decoding could distinguish the responses to these stimuli where we've avoided radial bias. The logic being that if brain decoding still works in this case, then that's support for the idea that it's not, or not just these coarse quadrant level biases that underlie successful decoding of orientation, but maybe that decoding really does exploit these subtle voxel by voxel biases of the sort talked about by Boynton, and potentially representing or resulting from just biased sampling of the local orientation map. And the result of our study was clear that even when we remove radial bias, we still find that classification performance is significantly above chance across the early visual areas V1 to V3. So this supports the idea that it's not just these coarse biases at a spatial scale greater than the size of a voxel that underlie performance, but that there are probably also more subtle biases at work that can drive performance. So, thus far I've talked about decoding stimuli, if you like. There's been no attempt to disambiguate sensation and perception. So what the subject is perceiving, the content of their consciousness, is what they're being presented with. Now, what we would like to be able to do is to dissociate sensation from perception, to study the, if you like, the private contents of our conscious awareness, dissociated from the sensation that gives rise to that perception. And so one way we can do this is by using ambiguous or bistable stimuli. For example, binocular rivalry is a paradigm where different stimuli are presented to the two eyes, and they result in a percept whereby they alternate for perceptual dominance. Typically when we present, typically our two eyes sample the world from slightly different positions, and so they receive different images, and these two images are fused to give us to contribute to our three-dimensional perception of depth. However, when the two images are sufficiently different, there's no way that they can be meaningfully fused to give us a three-dimensional representation. And under these circumstances, for example, when we have opposite orientations and opposite colours, then one of the images dominates and the other one is suppressed from consciousness. So we might have an unchanging stimulus, always having a leftwards green grating in the left eye and a rightwards red grating in the right eye. But what happens to our perception is that first we perceive one of these images, and then over time our perception alternates between these two perceptual states. So here we have a dissociation between sensation and perception with these purely endogenous perceptual fluctuations. So the question then is, can brain decoding decode the percept rather than simply the stimulus? Now, the first study to look at this was again by John Dylan Haynes and Geraint Rees, who also did one of the pioneering orientation decoding studies, and they found that when the classifier was required to discriminate the subject's percept, the subject monitored their percept by just pressing one of two buttons, then again they were able to get accuracies, these are again generalization accuracies, significantly above chance. So this is in areas V1 to V3. So it shows that brain decoding is able to discriminate not only different stimuli, but also different percepts of the same stimuli. An analogous study using a different different method of dissociating sensation and perception was carried out by Brewer and Van E a couple of years later. They used an ambiguous structure for motion stimulus. In this case, there's a set of dots on a computer monitor, and each of them moves backwards and forwards in simple harmonic motion. So physically, if we're viewing the monitor from the top, of course there's a flat screen, we've just got motion backwards and forwards, but what we see is a compelling three-dimensional percept of a cylinder, or in their case a sphere. And so this is the perceptive structure from motion. We see the three-dimensional structure purely on the basis of the motion of the dots. And this direction of motion, direction of rotation, is ambiguous. We get it's ambiguous as to whether the front surface is moving to the right or to the left. And again, as with binocular rivalry, we tend to get flips in perception between the two bistable states every few seconds, purely endogenous perceptual flips, so that sensation is again dissociated from perception. And what Brewer and Van E found in this case was that the early visual areas V1 to V3 weren't predictive of the subject's performance for this kind of stimulus, but that throughout the dorsal processing stream, areas V3A, V7, MT and so on, there was activity predictive of the subject's percept. So this is a generalization of the earlier Haynes and Rees result to the idea that classifiers can, if they're looking at the relevant part of the brain, they can decode perception decoupled from sensation. Okay, the final topic I'd like to talk about is the state of the art, reading the neural signature of consciousness. This is potentially very exciting, the idea that we can actually learn something about what makes a particular brain state conscious by employing this brain decoding methods. And this is of interest, of course, neuroscientifically, I think philosophically, and also it has a strong practical application, because if we have a patient who is unresponsive, clinically unresponsive, then it's very difficult for a neurologist, a clinician, to establish whether that patient is comatose or whether they're just locked in. That is, whether there is actually no conscious activity in the brain, or whether there is conscious activity but no ability to communicate, to signal that there is any activity going on. And anyone interested in this particular area of research, I would recommend the work of Adrian Owen and colleagues, reviewed in a recent Progress in Brain Research article. So I'd like to just focus on a very recent study, came out earlier this year, from Aaron Scherger and colleagues. This is one of the first studies to try and use brain decoding to actually establish the neural signature of consciousness. I think it's an exciting and ambitious study. I certainly think its results are subject to alternative explanations, possibly equally plausible in my opinion. But I think this is very much state of the art, and I'll just give a brief description of what the study's done, and then direct you to a couple of recent commentaries on the work, which, although written independently, actually converge quite closely in their viewpoints. So, what Scherger and colleagues did to, well, first of all, they presented subjects with stimuli that were the same in the two eyes, either two faces to produce the percept of a face, or two houses to produce the percept of a house. And they asked where in the brain was there activity that was able to discriminate faces from houses. And they found activity in the an anterior region of the temporal cortex. Now, they also manipulated the visibility of the stimuli by presenting the left eye's and right eye's stimuli in complementary colours. So there would be a red face on a green background in one eye, and the same face but green on red in the other eye. And these colours tended to fuse to give the percept of a uniform yellow. So again, these stimuli could either be both faces or both houses. In either case, the percept was one of a uniform yellow. So the subject in this case was unable to make the discrimination between face and house, hence the stimuli are termed invisible. And what they found was that there was a more posterior region of the temporal lobe that was now able to discriminate faces and houses. So it's surprising that there's this dissociation. There's a region that is able to discriminate faces from houses only when they're visible, but not when they're invisible. That's perhaps not so surprising, but the fact that there's a region that can discriminate faces from houses when they're invisible, but not when they're visible, is perhaps more remarkable. Now, when we look at the when they looked at the activity of this more posterior region, they found, this is really just summarizing, that when the stimulus was visible, the region was unable to make the categorization. When it was invisible, it was able to make the categorization. The other dimension that they looked on was the reproducibility of responses. And what they found was that in the invisible condition, although the area was able to categorize face versus house, within a category, the reproducibility of the responses was low. So we had quite a lot of variability in the responses to faces and in the responses to houses within category, even though between categories they could be reliably discriminated. In contrast, for the visible stimuli, categorization between the stimuli wasn't reliable, even though within each category reproducibility was high, i.e. variability was low. Now this is a surprising finding, and offers the possibility that what Scherger and colleagues were observing was part of the neural signature of consciousness. I mean, their title was that reproducibility distinguishes conscious from non-conscious neural representations, and they come up with some arguments as to what this might actually represent at the neuronal level, how it might come about that the conscious stimulus is represented with higher reproducibility than the unconscious stimulus. Now, I don't want to go into details here, as I realise that I've already talked for long enough, but I would certainly commend their recent science paper, and I've written a commentary in Current Biology, and Schwarzkopf and Rees have independently written News and Views in Science. So this is very much state-of-the-art stuff. I think it asks possibly more questions than it answers, but I think this is exactly the sort of application of brain decoding that's going to spur further work. And so I'm not going to finish with any real conclusion, because this is about the state of the art. It's not finished yet. We have evidence that we can use brain decoding to read, if you like, the contents of consciousness. We have some idea about what this might involve at the neuronal level, but that is an issue that is still being researched, still being debated, and there is the question of whether we can usefully employ this technique to actually learn something about what distinguishes conscious from non-conscious states. And while I think the jury is very much out, I am optimistic about the future of this new and exciting field. Thanks very much for your attention.